Yes. So today we are going to discuss on the topic that is genetics. So as in my last video you have seen that we have discussed on the things that is on the basis of genetic interaction. We have came across two words, that is intragenic interaction and intergenic interaction. So what is genetic interaction? The influence of allele or non-allelic or non-allele rather or non-allele or normal phenotype or normal phenotypic expression of genes. So what does this word mean? That is the influence of allele or non-allele, means two separate genes, suppose capital T and capital T and capital T small t. So these are the two alleles of a gene, and if I talk about this capital T and this capital T, then these two alleles are of non-allele, means they do not belong from the same gene. On normal phenotypic expression of genes, means they have an influence on the phenotype or on the phenotypic expression of both the genes. Or if I am talking about the two alleles present in a single gene, then we are talking about the phenotype of a single gene only. So let us see how many type of genetic interactions are there. Genetic interactions are of two types. What are they? Number one, intragenic interaction, intragenic or interallelic interaction. Now as the name suggests, intra means within a single gene. When I am having two alleles and both the alleles are interacting with each other, I am calling it to be a intragenic. So when two genes present on same gene locus, that is position, same gene locus on homologous chromosome, of a gene interact in such a way to produce a phenotype different from the type dominant or recessive. Yes, they are always this when two genes present on same gene locus on homologous chromosome of a gene interact in such a way to produce a phenotype which differs from the type dominant or recessive. Yes, as I said about codominance, over here you have seen that there is no dominant or recessive, but both the alleles are dominant in nature. Another one was incomplete dominance, where one of the genes or alleles present in a gene is not totally dominant. The recessive one has also an influence onto the dominant one or onto the phenotype one, and that's why Mendel had got pink color flowers instead of red, even though the dominant gene was present. Apart from this, under this example comes the multiple allele. Like the I gene has got three types of alleles, A, B and O. Now we are going to talk on intergenic interaction. What is intergenic interaction? When two or more independent gene present on same or different chromosome interact to produce a different expression. Means these are the two different non-allelic interaction. Non-allelic, as the definition of genetic interaction says, non-alleles means, as I say, two different gene alleles, this one and this one. Now over here, this gene and this gene, both are in same alleles, are in different genes. When they are present on same or different chromosome, they interact to produce a different expression. And the examples are epistasis, very very important for your exam, duplicate gene, lethal gene, complementary genes, etc. So these are the part, or this is the part that we are going to discuss today, that between when two alleles are present in two different genes, what do we see over there. So let us now talk about this. So this is what we have talked till now or discussed. Now we are going to start the topic, that is duplicate, or rather the lethal gene. Now what is a lethal gene actually means? Lethal gene, as the name suggests, gene which causes death to an individual, which causes death of an individual. When it comes in homozygous condition. Homozygous condition. So this is the first point. Second point is, lethal gene may be dominant or recessive, both, but mostly recessive occurs lethality. Next point, many of the genes which do not cause definite lethality are called semi-lethals. Example, coat color of mice, leaf color of snapdragon, and sickle cell anemia. These are the examples of lethal gene. Now let us discuss what this thing actually means. I will be giving you an example where you can see, suppose L. Cugnot was the scientist who had worked upon mice in the 17th century, where he had taken a mice that was having a heterozygous gene, and when it was crossed with a recessive one, over here this was yellow and this one was also yellow, both were heterozygous. So from here what he had got was capital Y, small y, capital Y, small y. So these were the gametes and this is the parental generation. So from here what he had got is capital Y, capital Y, capital Y, small y, you can see again, and the last one was. So this in F1 generation, what we had seen, that this mice dies. This is having yellow color, this is also having yellow one, and the last one is gray. So the ratio was always is 2:1. Now the thing is that, as you can see, let us discuss gene which causes death of an individual when it comes in homozygous condition. Yes, both the Y genes, and Y is the dominant one, and so this gene when present or when it goes to in F1 generation, if both the alleles are having capital Y and capital Y, then that is the lethal gene. Now this lethal gene, when it is coming from the parental generation in the form of gametes, and when both the Y's are going into a particular gene, what will happen? The rule says that when it comes in homozygous condition, so this is a homozygous condition, what happens? The mice in the embryonal stage only, this will die. In the embryonal stage only, it will die. It will never come to the population. Remember, the lethal gene will cause death in the embryonal stage only. In the embryonal stage only of the F1 progeny. Whereas over here only one of the genes are dominant and the other one is recessive, so that's why it is getting yellow, because it was yellow. And over here both the alleles are having recessive one, so it forms gray color, and it is also, this mice is also not causing death. So from here it's clear that lethal gene may be dominant or recessive both, but mostly recessive occurs lethality. But over here we can see that this capital Y and capital Y both are homozygous, it will be causing death. If both the recessive ones, if I had said that both this capital small y was a lethal gene, if small y over here was a lethal gene, so this would have been, or this mice would have died. But since I have talked over here, or I have said that capital Y is the recessive allele present in the F1 parental generation, so when both the capital Y forms a gene, the mice does not exist. Instead, in the embryonal stage only, it dies. So this is what actually we are talking over here. Remember, in lethal gene both the alleles should be in homozygous condition. It should not be that one will be in homozygous and in heterozygous, then it will not be causing death. It will be a semi-lethal case. So next, this was lethal gene. Now we are going to discuss on the topic that is duplicate gene. What is duplicate gene? Duplicate gene is also known as clone in genetics. Duplicate gene is also known as clone. The first point comes, duplicate gene or factors are two or more independent genes present on different chromosome, which determines the same or nearly same phenotype. So either of them produce the same character. I will be explaining it to you. Independent genes do not have cumulative effect, means additive effect also you can say. The third point is it produces the same phenotype whether present in homozygous or heterozygous state. As a result, dominant phenotype is abundant in F2 generation ratio 15:1. Over here we will be discussing on the fruit shape of, you have studied in the sexual reproduction of flowering plants, Capsella bursa is of two types, triangular bursa pastoris and the other one is ovoid. So now let us discuss what it actually tries to say. Actually, it is talking about, suppose this is a homologous chromosome, over here it is having gene A or allele A, and over here this is a homologous chromosome which is having a dominant gene B. So both the genes will never be combining together to produce the same effect. They will never be working together. They will always be occurring individually. They will be representing or causing the effect individually, and each one may be in the different gamete or in the different gene to produce this effect, but they will never be coming together to produce the same effect. This is what cumulative effect. Cumulative effect means additive effect. The plain simple knowledge about duplicate gene is the additive effect, where one gene does not work with the other gene, means like A will never be working with B or B will never be working with A to produce the same effect. They will be working independently. Let us take the example of, suppose capital T, this is a type of gamete which is totally dominant, and this is a type of gamete. So from here in the parental this was the parental generation. In the gametes, what we will be getting? We will be getting T1, T2, and from here we will be getting T1, T2. So if we do the cross of this, what we will be getting from here? We will be getting, if this is a checkerboard, we will be getting from here T1, T2, again it will be T1, T1, or rather T2. After this we will be getting T2, T1, or rather, instead of doing it in this way, let us discuss in this way only. One will be T1, another one will be T1.
Yes, yes.
Another one will be T1. The next one will be T1, T2. Another one will be T1 and T2. and the last one will be T1, T2. So this is the type of gamete we are getting over here. Same thing will be over here. T1, 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 T2. The next one will be T1, T2, and the last one will be T1, T2. So when we are forming the gamete, what we will be getting over here? We will be getting over here, remember what it says, that it should be dominant. It says when present in homozygous or heterozygous state. So see, out of this two, T1 and T2, T1 and this is T1 and this is T2, what we will be getting, or rather instead of this, let us discuss in this way. Remember that when these two gametes and this one will be combining, always what you have to get out of T1 and T2, always one of the gametes should be, that is T1 and this is T2, the other one will be also T1 and T2. So when such type of gametes are there, we call this to be as, suppose I am representing this as a round one and this one to be as a ovoid one. So all will be round over here. The next one will be what? T1, T2, T1, T2. So this one will also be round over here, means out of this four alleles, if any one out of T2 and this T2, which one? There is a dominant allele present over here and this one is also a dominant allele. Both are in homozygous. So obviously it will be round. Means you can never say unless and until it's T1 and T1 and it's T2 and T2, then only we will be getting a ovoid shaped flower of capsula. But other than that we can't say, because over here both the alleles of T2 are ovoid, but over here the shape of this one is having one of the dominant alleles. Like gene A and gene B, as I said, both needs to be present together in the gamete. It cannot be like this, the small a and capital A, and this is capital B and small b. Both will not be required together to produce the same effect. It is not mandatory. They cannot require, we do not request such type. It will be producing individually. So therefore, what happens if any one of this is present in the gamete, then it will be a dominant one. As you can see over here, this is causing a dominant gene and T2 is a dominant gene. So unless and until if any one of the gamete alleles is a dominant over here, we will be getting it to be a dominant one. This is what duplicate gene is all about. We will be taking some few more examples based on this. The best example over here is, as I said, this one was the best example. Another one more example I am giving you, that is, suppose I have taken capital A and small a, and I have taken capital B and small b. So from here what I will be getting? I will be getting capital A, capital small a, capital B, small b. So what I will be getting from here, capital A and capital B, and this one will be capital A and small b. This will be capital B, small a, and the last one will be A and B. So remember, over here both the alleles will be representing it individually. Suppose A represents round and B represents ovoid. So what I have told you, that when they are present individually, like A and B, over here it will be the round one, over here this will be the ovoid one, and in A and B it will be a different one. That is the recessive one, which I have said over here as 1. Now A and B, when it will be present over here, both round and ovoid will be expressed in this gamete also. It is not like that both will be mixed together, because it says independent genes. They do not have cumulative effect, means additive effect. It will be expressing individually. half of the seed will be round, half of the seed will be ovoid. It is not like that it will be having some different shape. No, it will be the dominant one, and both the alleles are dominant, so it will be expressed in individually, whether in homozygous condition or in heterozygous condition. A single dominant allele or gene can express its character. So that's why dominant phenotype is more abundant in F2 generation compared to that of recessive one, and that's why the ratio will always be 15:1. I have taken a simple example from here. So that was the thing. If you have crossed this, if I had crossed it with, suppose from here, capital A and capital B, the next one is capital A small b, after that capital B small a small b. Over here capital A, capital B, capital A small b, capital B small a, small b. This would be, next one would be, then it would be so as you can see, this will be a dominant one. Both, all the alleles are dominant. This one will be a dominant one. This one will be a dominant one. This one will be a dominant, because over here see both the alleles are present. This one will be a dominant one since one of the gene is dominant. This one will be a dominant one. This one will be a dominant one. So wherever, if any one of the allele is also dominant, then it will be expressing a dominant allele only. So all over here are dominant, because each one can express individually, even if one of the allele is also present, either of A or B. So this all are dominant. So count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and this one will be the recessive one. So the ratio is 15:1, because till now before duplicate gene we have heard that if any one of them is, Mendel said, if it's a heterozygous, then it will be not a pure dominant. So but over here, since A is the dominant allele and it is able to express, so we are calling it to be a dominant one. Even if both the B genes are recessive over here, it will be having a dominant phenotype. So this is what duplicate gene is all about. Remember, it is not a cumulative effect, it's the individual effect of both the genes. Now today's last topic is complementary gene. Complementary genes.
Oh, হাত ধরে গেল রে।
বলছো কেন? ও